Hello, this is the Victory Podcast, and I'm your host, Monique Watson. This is another installment of our COVID-19 series. On this episode, I sat down with my sister, Nicole Creppel. She's a fourth-year medical student, and we talk a bit about her professional background, as well as some of the research that she's involved with now. We also talked about how viruses work, how especially this specific COVID virus works. Additionally, we also talk about the implicit bias that exists in the medical community and how that has an effect on everything from medical decisions to clinical research. Let's listen in. So, welcome to the Victory Podcast. I'm your host, Monique Watson, and I have the wonderful pleasure to have someone who I've known literally my whole life and her whole life, Nicole Creppel. How are you doing, Nicole?
I'm doing well, doing really well, trying to stay indoors more and enjoy this holiday season as best as I can. This is our first Christmas that we haven't all been able to be in town in like a long time. So
yeah, I think it's been like, I was looking at, Google, and Google Photos will tell you like this time 6 years ago. I think 6 years ago was the last time we both weren't home. So for our listeners, Nicole is my sister, 100%. I mean, we haven't gone on Maury or anything to do a DNA test, but I'm pretty confident that you're my sister. So maybe, so just let me first give an outline to the folks listening. So how we're going to kind of go today. So we'll learn a little bit about Nicole's background, some of her professional background. If you heard a little bit is our personal background. Also, we'll be talking a bit. This is part of our COVID-19 series. So we'll talk a bit about viruses, which is what is the disease causing agent of the COVID-19 illness. Talk a bit about viruses, how they work. Information about the vaccine. Nicole will give us try and simplify a bit about the vaccines, how they work in general, and some of the current vaccines that are out for this COVID-19. And then we'll talk a bit about clinical trials. There's some questions that have come up in our earlier COVID mini-series around the trials related to the vaccine and the diversity or lack thereof, and kind of talk about some of those challenges, both in this maybe that more apply on a greater scale, and then implicit bias in the medical system, a little bit about that and sort of what are certain things that you're doing as an individual and some programs that you're helping to put together. And then close us out with info and how you can stay in tune with what Nicole is doing. So, Nicole, tell the people.
Hello.
They don't know you like I know you. So tell the people a little bit about yourself, some of your professional background, kind of what got you from here to there.
Okay. So I am currently a fourth-year medical student, although I am doing a year of research right now. I go to Meharry Medical College, which is in Nashville, Tennessee. It's a historically Black medical school. And actually, our president, Dr. James Hildreth, he's actually an infectious disease specialist. So he's actually played a major role in Tennessee for helping people and spread information about this pandemic and everything. Currently, I'm doing research at UT Memphis, and I'm doing some of my research is revolved around, we have a COVID, like a droplet study and studying droplets and things like that. And I have a project with creating this diversity and inclusion program and other little things that just pop up here and there. I'm working with the Department of Otolaryngology there, which is ear, nose, and throat. And so before that, I have a background in biochemistry and molecular biology. That's where I got a master's in from Tulane University. And before that, I did something completely unrelated, which but did give me a great chemistry background. Is at Georgia Tech, I got a degree in polymer and fiber engineering. So and also currently I'm enrolled at Johns Hopkins in their master's in healthcare systems engineering. And so that's a recent interest that I've also taken up because of the systemic changes that really need to happen in healthcare.
Wow. That's a lot. I honestly, I mean, like I said, she's my sister, but I don't always know all the ins and outs of all that background. So I think you're uniquely qualified to talk about all the things we're going to talk about today from the different facets, right? So let's talk first. I'm interested, first off, so in your studies that you're currently like hot off the press research around the droplet study related to COVID. So are we analyzing the actual like droplet and what they're composed of from people's respiratory droplets, viral load, kind of what is that? As much as you can share, I know that, it's active research going on.
Yeah, sure. So we have a joint project, University of Tennessee with the University of Memphis, that we are working with speech language pathologists. And it is actually studying the actual motion of droplets. We're using laser technology to be able to detect the trajectories of droplet spread and being able to measure the amounts with certain words and certain activities with your voice and singing and different things like that. And from there, we'll probably pilot to a bunch of different looking at other things further. But right now we're just trying to get an idea of can we detect these droplets and how many droplets come out when you are doing certain activities. So, yeah.
Wow. I've seen some similar research. Absolutely. Yeah, that sounds like very interesting. So we'll have to stay abreast of as that comes out. Okay, so let's talk a bit about, so we're, as we're recording this, FYI world, we are in the midst of a global pandemic. In case you've been on some remote island for the last 9-10 months, it's a whole thing. So let's talk a bit. So COVID-19 is the disease for that is created by
SARS-CoV-2.
So SARS-CoV-2 virus. What do we know about viruses? This virus is sort of how viruses work, right? Are they alive? There's some discussions in the lay world about whether viruses are really alive or not. Maybe you can talk a little bit about that.
Yeah, so I can give like a brief kind of rundown about viruses. I think when people think about viruses, since we can't see them, people just think, okay, they're like bacteria. It's just a different type of thing, And in a way, it's kind of similar. Viruses are technically not considered life by the definition of life, meaning that they do not replicate on demand autonomously. Like they don't have every single part of the machinery that's required to duplicate itself. And so that's why they say that they are technically not alive, but they, with their interaction in the environment, they are very, very, they're not dead either. They immediately can function as long as they stay in their environment that they're able to thrive in. Viruses have two types of genetic material. Some of them have DNA, just like me and you have DNA, and some have what's called RNA, which is basically like DNA, but it's just single-stranded instead of double-stranded, and it's got a little bit of different shape. But we also have RNA. We just have RNA that comes from DNA, and that is a whole process, and I can go into it, but that is how we get the materials and the proteins and everything that our bodies make is from RNA, giving this set of instructions, if you will, to our body. And this process happens, all of our different cells. They all have like our cell. If you remember basic biology, they have the nucleus in the middle, and that's where our DNA is. And then there's the cytoplasm, which is where all of our machinery is. So we have our nucleus and we have the cytoplasm, and that's where all of our machinery is. That's going to tell us what to make proteins, what to get rid of, telling us if there's damage, all of those different things. A lot of things happen in the cytoplasm of the cell, which is, if you're looking at an egg, it'd be like the egg whites. The yolk would be like the nucleus. That's where all the genetic material is. With that being said, so this particular SARS-CoV-2 virus, its genetic material is made out of RNA. It also, because it does not have its own machinery to replicate itself, it needs to live inside a certain type of cell. And what they do is when they enter our cells, they hijack our machinery. They hijack our like functions or whatever, host, animal, whatever, and they hijack their machinery so that they can get the proteins that they need to make more particles. And this is a cycle that starts where they just attach to the cell, and then they trick the cell into what's like engulfing it, basically. And they go into this particular RNA viruses, because coronavirus is an RNA virus. That's its genetic material. They replicate in the cytoplasm. This particular strain is immediately ready to start using our machinery without having to make any type of changes to itself or anything like that. So as soon as it enters, it can start hijacking the machinery of the cell in order to start making proteins, which give instructions for the viral particles. And then they put all within our body, they'll make the proteins and then like put it all together and make new virus particles. And then those particles get released and then they infect new cells. And so you see how this cycle begins and how it can easily just invade cell after cell after cell after cell. So that's kind of viruses in general. And that's kind of this particular virus, right? And so when our bodies, we have a way though of kind of doing a check. Like our immune system kind of has a check on itself. And when periodically we have these things that basically present the stuff, the proteins that are in the cell, and they kind of like bring it up to the surface and let our immune system check it out. They're kind of doing it's almost like a TSA type check. Or surveillance type check. So while
it's we like have a random security check.
Yeah, random security screen. So they like just bring up a random protein. They're like, hey, this is what we're making right now. They bring it up to the cell surface, and then your immune system says, is this self? Like, is this my normal proteins, or are these proteins foreign to me? Are these proteins that like are like some type of foreign invader? And so that is how the body protects itself. And when it detects an invader and it bonds to it, and it's like, I don't know you, but I'm going to remember you. I never forget a face is basically what they say. And they start making what's called like antibodies, and they send an alert, and they're like, hey, they basically put like BOLO signs everywhere for like your whole immune system.
And BOLO, for those who don't watch all the criminal shows out there, is be on the lookout. So your body is looking out. We saw this virus, wanted dead or alive type thing.
Right. And so once your body makes these what's called memory cells or memory cells, if they ever encounter this, junk again, like this type of protein that has that structure, then they are going to immediately recognize it. This process in the body takes about, let's say roughly about 10 days to happen from the period of when it recognizes it and it makes like the proteins to remember it and everything. So it's not an instant process. We have another immune system that kind of just does generalized killing for that, for the kind of our immediate responses that we have. So that's a little bit about our immune system and how they respond to viruses. You have any questions about any of those things? Or
so let me just make sure to try and understand completely. So viruses aren't technically alive because they can't make themselves by themselves. They require hostile takeover of our cells to replicate, to then create more of themselves. Our body can kind of memorize what that virus looks like. It's like, oh, I've seen you before, and they over a period create these memory cells to that way if it comes in contact again, it can take it on and basically go after it to eliminate it. Mm-hmm. Right?
Yeah. And so, actually, if I can interject, I meant to say, the viruses, they have different properties. Some viruses have to live in like a certain environment in order for them to even like maintain their structure and everything. And this particular virus is an enveloped virus. And so that envelope is like kind of a bad thing because it makes them really susceptible. That envelope makes it so that they can't, they dry out and then they will like die. So like die in the terms
from a functional
of,
sense.
Yeah, from being able to not be able to make itself anymore. And so they're susceptible to drying and things like that. So that's why they have to live within a droplet in order to maintain the ability to replicate itself. Whereas some viruses that are naked, they can attach right away and they don't need to be in a droplet. They can, live on surfaces and things like that. So that is one good, or they can live in the air even, and that's one advantage that's good with like coronavirus is that it requires a droplet. Whereas if it was something that was airborne, and by airborne meaning that it doesn't need to be in a droplet at all, then that would be like even worse.
Challenging.
More challenging to control.
Yeah. So I think it's a good overview for folks. Of course, reach out to Monique at the Victory Podcast if you have questions, want more info. There's a lot of good information on CDC and WHO that can talk about viruses in general, as long as a number of other like peer-reviewed sources that I'll try and link in the show notes so people can have that information. So we have these viruses. People's immune systems are responding to them. There's sort of been not much until you get to this sort of vaccine stage that we've now arrived, that there is a credible vaccine. And sort of maybe at a high level, we can talk a bit about, how do vaccines work? Why are they important? And specifically, maybe if you know anything about these, either the Pfizer or the Moderna vaccines.
Okay, so as I mentioned, in order for a virus to come in and hijack the cell, there's an interaction that happens on the cell surface between the protein that is on the outside of a virus. And they connect with each other. They make a connection, and
between their cell and the virus, right?
They make a connection, and it kind of unlocks a key and says, "Hey, let me in." It tricks them into thinking that they're supposed to be in there. So these cells enter that way. So all of the whole idea of vaccines and things like that are to, of these vaccines especially, are to stop that interaction from happening, and not allowing it from being able to enter. So a lot of those types of solutions are going to be what is going to be our best attempt at having that interaction not be successful. And so they try to exploit things that are different from humans' cells so that it can target things that like certain properties that are unique to viruses. And so that's kind of the whole idea between any type of drug or vaccine targets is what is something that's unique to viruses so that when we're targeting it, we're not killing human cells also along the way, if possible. And this particular, so there's a few vaccine candidates out there. The ones that are approved right now, I can explain very high level of basically how they work if you would like.
I think it would be good just for folks to kind of understand. And I'll also share info. So if people kind of struggling visually to understand, we'll share the CDC info on it as well. But I think a good high level, like how does it work?
Sure. So what this thing does is the vaccine is this particular type of vaccine. So traditionally when people make vaccines, what they would do is they would like grow a virus. They'd have they'd start with a virus sample and they would grow it in the lab. And then they would be like, okay, how do we grow a certain protein? And they'd test it and things like that. Right. And so they'd have to start with the virus. It takes a lot of time to grow in the lab and to do the right analysis and everything like that in order to do that. But what's really cool about this particular technology used in these vaccine candidates that are approved right now, they actually use a part of a part of the genetic material of the coronavirus. So that RNA that we talked about, the mRNA. And what they do is they just use the genetic material, they package it into a fat bubble. And they use a fat bubble because fat goes into cells easily. So they package the DNA of the virus, but only the RNA of the virus, but only the part of the virus that is responsible for like making that connection to the cell. So on the coronavirus, that's the spike protein. And the spike protein interacts with the cell. I think there's like maybe 3 or 4 different spots that it can interact with. And so they found all of those sequences, and they just specifically to those, packaged those together and then put it in a fat bubble. And then they have that enter the cell. So it almost looks like a fake virus almost, but it's not, it's just like a fat bubble. And then it just has a piece of genetic material. So then once it goes into the cells in your body, what it does is it goes in and unpacks itself just like anything else would. And it goes through that genetic material, uses your machinery, and uses the machinery only in the cytoplasm. It's not using your DNA. It's only using the things that make protein, which are called ribosomes. So that RNA sequence is going to be processed through your ribosomes in your cell. And they're going to make a protein, just that protein, the things that are in that RNA strand that we told them. It's not making a whole new virus, it's just making a protein on the virus, very specific. And what it's going to do is how I told you, how they present what's on the menu kind of thing, and they have them random security checks. So what's going to happen is that spike protein is going to be presented, just because that's what the cells do. They are always presenting what they're processing. They are going to present that to the cell surface, and it's going to force your immune system to be like, hey, you're not one of us. There's that jerk. Remember his face. There's that jerk. Remember his face. I'll never forget a face. And they start putting the bolo signs out. Essentially, that's kind of the whole, that's the whole thing. You're just getting the cells in the body to make that protein, which the protein is completely harmless by itself. It's making that protein, it presents it to the cell surface, and the cell is like, oh, hell no, we're going to remember you, and we're also going to kill you, because there's 2 types of cells. They have the ones that kill, and then they have the ones that, tell you to make more of the immune cells. So they have 2 different types that get that up there. And so they're going to get killed too, because that's part of the job of your immune system. So, but in that period of time, so let's say you get the vaccine, remember I said it takes about 10 days for those cells to make. So, that's where the body starts making those cells, and then they'll keep making them, those memory cells, and then it'll keep making them and keep making them. And your body gets a chance to just get better and better and better at attaching it. So then if it ever comes encounter with that virus again, it's kind of the same thing. It's like, oh, I don't forget a face. And then your body immediately recognizes it and can respond right away. And the more times that the body's exposed, the better it is at responding, or if you are exposed more than once. So that's why you have that second shot in there, to give your body a chance to be exposed again, and it'll make even more cells. So that way the delay between when the virus attaches and when it has the cells ready to act on it is like much, much shorter. So that's what makes the vaccine effective, is because it's able to act before the virus really gets a chance to start taking hold of the cells, and before it gets the chance to start It needs to act before the virus does its thing and tricks people. So people's cells. So yeah, that's kind of in a nutshell about how this particular vaccine works.
Yeah, what are you seeing? Because I know you've done some clinicals a bit and some different rotations, and sometimes even in your studies starting out, hesitancy around vaccines and sort of what are you seeing there? And then if there's we've talked a bit on this show a bit about hesitancy specific to this vaccine, people's questions about it, sort of some people's concerns center around the speed in which this was created compared to normal vaccines or medicine kind of trials and all that kind of stuff. And secondly, if you have any comments there, if not, that's fine. Secondly, the also lack of diversity in some of the clinical trials. We talked a bit about this with Dr. Bent, who is our aunt. So we talked a bit about that. She had some concerns of which she understood the challenges of that because of the for several reasons about lack of diversity and challenges there. So maybe we can talk a bit about some of the concerns that you have heard, seen around vaccines, and even more so if you've heard anything specific to this vaccine as it comes out.
Sure. Yeah. So, the anti-vaccine effort in general has existed for a very long time. I mean, back to 1802, there was the anti-smallpox. That was the first cartoon that showed humans growing little parts of cows for if they got injected. So there has been always a fear of the unknown, pretty much. So even to now, there's a huge, very well-funded, very star-studded movement that is anti-vaccine. And a lot of times these people, I can't quite talk about their intentions or whatever, but they use information that's not completely correct. And I don't know if it's based on their limited understanding and things like that, but there's a lot of anti-vaccine messaging that is on the rise. And that has a lot of support from people, but a lot of it is not based entirely on the truth, and they're based on some things that are like very skewed or very poor studies and things like that. So I can't remember what was the second part about your question with the with this vaccine. Oh, with the amount of time.
Speed. Yeah, the speed in which it was developed.
One of the things that has come up, and they're like, heck no, I'm not taking this. It's only been so many months and they already have a vaccine. It's already been tested, all of these things. Right. And so that is something that comes up a lot. The thing is that the technology for this vaccine has been happening for it's been developing for a very long time. Normally, to get the process of getting to a vaccine, it has a lot of there's like a stage where where they obviously make the discovery of what is like there are scientists out there that find viruses that can potentially infect people and cause problems, and they start doing research on what are some things that we can exploit. What are our vaccine candidates? So there's a whole process in determining like specific vaccine candidates and what are good candidates, meaning like a good candidate is something that is unique to that virus, that's different from human cells, that'll allow the body to mount a response against it. Something that's going to be obvious enough for the body to pick up and become and mount a response specific to the virus. And so there's usually a lot of that. And so some of that's a lot of back and forth, and they do a lot of analysis and things like that. But the good part, and then once they figure out what's the best one, they try to get funding. So they have to get funding And a lot of times they have to get a lot of support from different investors and really kind of defend why this is the best candidate over another candidate. So there's a lot of that back and forth. Now, this process for the coronavirus has been happening for about almost 20 years, at least, because with even before SARS, but with SARS and MERS, this was a really big push.
And so SARS, just for context, SARS and MERS were earlier. Like we saw, I think, a lot of SARS in both the Middle Eastern regions and Asia, China, those parts of the country.
So SARS
similar sort of. Go ahead.
SARS is another coronavirus. So this is SARS-CoV-2. That was SARS-CoV-1, and that was back in Asia, and they had a lot of an outbreak then. And so that was one thing, and then MERS was Middle Eastern. SARS stands for what is it, like severe acute respiratory syndrome? MERS is Middle East, and then MERS is Middle East respiratory syndrome. And so so that's what and that's the worst case scenario for these coronaviruses. And so when SARS hit, people were scrambling then, and they were like, we need to get this under control. What are like the vaccine candidates? And so some candidates were actually presented way back then, and they figured out from there, like, what are the best ones if there's ever another coronavirus? Like, what's going to be our target? Like, if this happens again, what's going to be our target? Like, what are the ones from our studies that we did then? What are going to be the things that are going to be the most effective for that? So with that information that they've known since SARS, once they isolated that whole process, that's usually the longest part of developing a vaccine, of that back and forth, that was kind of like gone, because they already knew when coronavirus 2 hit, they already knew what they were going to be targeting. All they needed was for someone to isolate the DNA that is specific to that. And
RNA,
the RNA, the genetic material that is specific to that virus, for that protein. They're like, okay, we want the spike protein. What part of the RNA is for the spike protein? We will duplicate that part of that, and we will make it. And so once that technology was created, that was easy. And then the resources issue, I mean, obviously there's a pandemic, so the money was flooding in. There's lots of people that they had like an all hands on deck type of approach. And because usually vaccine development happens in a series, right? And these steps happen in a series, but there was a lot of things that were able to happen at the same time because you had multiple hands on deck. Like, you had a lot of infectious disease scientists abandoning their bread and butter research and directing their entire focus on this virus and like on creating things for this vaccine and developing these things. So you have the people doing setting up the logistics and everything for the studies, setting up, like, they have people that design these trials to make sure that they are going to be effective and they're going to, be valid type of studies. And so you have that going on while they're also growing the virus. Replicating the parts needed for this vaccine. So that just saves a tremendous amount of time, and just having the money, you're not needing like to go back and impress like any type of donors. Like, the money's there already, and you just be surprised at how fast things happen when you have the money to get them done. It really is a very quick process. In order it was a quick process for them to design the actual vaccine, because they already knew what they were going to be designing before they went into it. They just were customizing it, if you will, to this virus. It's like having a prefab kind of house, and then they customize the fixtures, if you will, for the house. So that cut down a lot of time. And then other thing that cut down a lot of time is that once they knew that it was safe, after the first data came out, the nation already had committed to like pre-ordering these vaccines. So once you start doing that, I mean, that also cuts down a lot of time, because they kept the important steps, which are the regulatory steps that are needed, but they cut a lot of the bureaucracy out, a lot of the back and forth that happens, and they streamlined a lot of those efforts so that they could do so that the bulk of the time that is needed to develop this vaccine was pretty much only focusing on if it's going to be safe and effective, its safety and efficacy. So that was like the bulk of what took the most time. And then everything else, they had it streamlined so that it's like, as soon as you can say go, it's ready. You got the orders filled, you got it, it's in the pipeline. They have prioritization for the FDA to review. They have all of those things. They put everything else on hold so that they could do this. So understandably, this is why they were able to be so efficient and so quick to be able to develop that, because these trials actually they're just like any other trials. So the Pfizer vaccine trial was 43,931 people that were enrolled at 150 clinical sites in 39 states. The Moderna was 30,000 enrolled, 89 clinical sites in 32 states. And that's really, really important, because the more people that are involved in a clinical trial, the more you can say that their results are valid, because you've sampled more people. You've been able to, the larger the groups, then the more you can say that these findings are applicable to the general public. And that's also why the racial and ethnic distribution thing is important, because people also want to be able to say that the studies are valid in all of the populations of the US and not just certain populations, because different people have different, properties about them. Some people have more severe disease, different cultures, different comorbidities, different things like that. But by race, socioeconomic status, all of these different things play a role in our overall health. And so they want to make sure that they do that. Now, one of the things that also comes up is people were like, oh, well, there weren't a lot of Black people in the studies. There weren't a lot of Hispanic people in the studies. There weren't a lot of this and that. And that's actually false. So typically there is a very low amount of people from minority backgrounds that are involved in clinical trials. And this is very, very true. But for this one, there were so many partnerships that were developed that the actual racial and ethnic distribution of these studies very closely mirrors the general population of the United States. So for instance, the Pfizer vaccine, the racial distribution is 13% Hispanic, 10% African American, 6% Asian, 1% Native American. 45% of the participants were ages 56 to 85 years old. For Moderna, 20% were Hispanic, 10% were African American, 4% were Asian, and 3% were all other races. And then 64% were ages 45 and older. So it's really good, because even though, yes, the minorities, they were all majority of the people were white, but the makeup, the representation mirrors that of our population. So that is a good thing, because that is something that is showing that there was some amount of diversity that is expected, where everybody was pretty equally represented as far as per capita, per population that exists in the United States.
Now, do we have any, you mentioned about comorbidities, and Dr. Bent talked about it in part one of our series about, things like, respiratory disease, kidney disease. I'm just going to use general, organ system diseases, not to get too granular. Do we know anything, or have you been able to find about the percentage of folks of those studied? I don't know if this information is out there, so the answer could be, we don't know. But how many, if any, of those folks also had some of those comorbidities?
I'd have to look up the actual, what was the inclusion and exclusion criteria. I do know that one of the things that was excluded were pregnant women and people who have experienced any type of anaphylaxis, which is like a severe allergic reaction that requires an EpiPen for any type of like drugs or anything like that. So they are excluded people like that because they wanted to make sure that they didn't trigger, something, and they could really isolate that. They actually are starting to I know they're starting to focus on like certain comorbidities and things like that, but I think that it was a lot of people just in the general population, and as long as you weren't like, I think, immune compromised, or if you didn't have anything that would actually like something to where like your immune system doesn't work, so they can't like really test the efficacy because your immune system's broken. I think for the most part, and I can double check, all of the clinical trials, even if they're closed, they're available on clinicaltrials.gov. And so you can actually look up the different criteria for that. But from my understanding, they were, as long as it was deemed a safe for them to participate, like as in they didn't have any type of really, really huge issues, then I think they were allowed to participate. But I don't want to misspeak.
Yeah. So you were talking also a little about side effects, and some of the questions that sometimes comes up, I think, is if they've either had COVID already and got over it, sort of some of those things, some of the, as compared to side effects from the vaccine itself, and some of that. What are we learning about kind of like long COVID or post-COVID effects? If people are like, I'll just get COVID and beat it and be fine.
Yes. So one of the things that's a really good point is, and it's a very, when you're evaluating like whether you want to get the vaccine and things like that, like that's a really valid thing to wonder about. And I will say that having COVID is so unpredictable. This virus is for it to be such a simple virus, it's so freaking smart because it can infect so many ways that like you really can't tell like what you're gonna have. You don't know if it's gonna be severe. You don't know if it's not. It really just kind of varies from person to person. And a lot of it has to do with how much you've been infected with the virus, like the load of the virus that you're taking in, and how long it takes for like your body to build up enough immune cells. And basically can it play catch up? Because so, and that's another thing I guess to bring up really quickly is that people talk about like not wearing masks and things like that. And, people are like, oh, masks don't work. But there's a minimum infectious dose that has to happen with COVID. And it's pretty low for COVID. I think it's only like 10 particles. But if you can reduce the number of particles that you take in, the virus replicates on a certain timeline, right? And so if you start with a lower number to begin with, and so there's fewer particles in your body by the time it like your immune system sees it and everything, and you have cells for it. Whereas if you start off with like 100 viral particles, or you go to the store, you get infected by one person, you get infected by another person, you're getting infected all around. One of the things that people don't really appreciate is that, it's one thing to get infected by one person, but when you get infected by like 10 people, that's just a bigger load of virus that you're carrying. So that's even more catching up that your body has to do. And if it can't catch up in time, what's going to happen is your body's going to be like, oh, shoot, like time to go into overdrive. And it's going to release everything that it's got. And a lot of these complications are related to what's called a cytokine storm, where the body is basically grabbing for straws, and it's just going to release everything that it has. And that is what kills people with COVID. And there's some other things that are happening too with when you actually get infected. Let's say that your body does catch up and it does mount immune response. and you beat the virus and things like that. But you were infected with the entire virus. So all of the components of the virus. So different types of parts of the virus were presented to the T cells in your body, to your immune system, and
to your TSA checkpoint. That's the T cells are
TSA.
like, hey.
Exactly. So you had different parts of the virus that were presented to your immune system, and no control over what parts of the virus were brought up or anything like that. And so so the things that your body makes the memory cells to, they can just vary a lot more. And some of those things may have like too much of a similarity in structure to like existing things in our body. So sometimes that can, that's how some people end up having like these like autoimmune types of problems and things like that, because maybe something that the memory against resembled too similarly to like a tissue that's in our body. And so there's things like that. Like that's a theory that like can be or related to that. And so I think that it's something to really consider, is that it's very individual, and it comes down to your immune system and the fact that because this is a virus that, so like the same thing, like people are like, oh, well, I get a flu shot. I don't get a flu shot and I'm fine. So there's a difference between, just like there's a difference between seasonal flu and like a pandemic flu, there's a difference between seasonal COVID, because there's COVID that flies around normally. I mean, not COVID, there's coronavirus that flies around normally and just causes a common cold. And there's this coronavirus. Okay. And the difference is that when a virus like this one, and like certain flus, like in the Spanish flu, when it's a virus that is not usually infecting humans, and they gain the ability to infect humans for the very first time, the humans do not have a way to have any like mild type of way to respond to it. So it's very, very devastating, versus if it were a coronavirus strain that normally infects humans, then it would be fine. Like we're the natural host, and it's adapted, and it's less of like a toxic, like you would get sick, but you would just have like a common cold. Same thing with the flu. Like you would have some people that have a level of immunity to it. No person has any baseline level of immunity to this strain, because this has never been in humans before. Just like no person had immunity to the swine flu because it was from a pig before. So it's the same type of deal. And when you don't have any type of baseline immunity, that's what it takes a lot longer for those cells to be developed, because it's starting from scratch. It's like making pancakes from scratch versus Bisquick or something like that. And so it takes a little bit longer, and the immune system doesn't have as much practice with a similar enough particle to be able to readily identify it as much, even though it's very good, but it takes a little bit longer. So that's why it's really important to really consider like the fact that it really depends on how much of the virus you're getting infected with at a time, and your immune system response. And that's why it's not just elderly people that can die from it, and it's not just immune compromise. It's just that it happens that elderly people, everything moves slower in elderly people. In immune compromised people, their immune system is suppressed. So it takes them longer to build those cells. So that is why they're more susceptible. But if you are so far behind to begin with, even if your body does build the cells, it's too far behind, so you really want to limit the amount of viral load. That's why some of these doctors would die that were very healthy, because they get infected multiple times by multiple patients, different things like that. So it really does matter. Like it's not just you get an infection and that's it. Like the quantity of it also matters. It matters what tissues it's invading, and it matters all of those things. And so that's something that people need to understand. And it can affect multiple systems. It does affect clotting because your body goes into this like inflammatory state. And when you're in this inflammatory state, you are more susceptible to clotting because your body thinks there's a bobo somewhere and it needs to clot and things like that. So problems associated with actually having the infection and that type of response and the damage that virus is like hijacking of your machinery, that related to there being so many particles hijacking at the same time and things like that. All of those types of side effects are huge problems, and there's no way to control that. Is much worse, much more grim
than any from the vaccine.
Than any vaccine. That's just something that I wanted to just also make very, very clear, that even if you don't die, and even if you don't die from COVID, there are long-term effects that you have less control over than with a vaccine. Because with a vaccine, they have already parsed out, like, this is the best candidate because of the low risk of it being similar to something in your body, like because of all of these different properties of it, because of the ability of the immune system to mount a strong response against it, things like that. They've already basically prioritized the best component of the virus for your body to learn and everything. So you have a lot of quality control. So there are fewer side effects. Any other side effects that have to do with the vaccine are that are unique to the vaccine are only short-term things or related to an allergy. And so that's why they made this vaccine very, very clean. And it's actually probably going to serve a model upon which other types of vaccines can also be developed and refined for.
Yeah, no, that makes sense. So maybe we can talk at a greater sense, and you've done some work and study and some courses around some of the challenges in general in clinical research, whether it be trials or otherwise, but clinical research and getting a diverse population. What are some of the things, either from the course you were taking about this, or that seem to be some issues? And then if there's some, people designing clinical trials or thinking about participating in one the next time when this is all over and they go to the doctor and someone says, hey, we'd like you to participate in X, Y, and Z. What are some of the things that you're seeing there as to why they run into issues in having enough visible diversity or diverse populations within their clinical studies, whether there be your droplet study or what have you? And what are, if there's any tips and tricks that you've learned as far as what people in that field could do to make things better for folks, to make them more likely to participate, if there's anything that they could do.
I gotcha. So in general, the clinical trial participation, 1 in 5 clinical trials actually end early for failed participation, or they complete with less than 85% of their targeted enrollment. And so this can be a bad thing, because that means that it lowers the power of your studies, meaning that you have a more limited understanding of what are some factors that might have taken a role to influence the outcome that you have. So there's certain confounding factors like that. That means that there's more limited information about the safety and effectiveness across different populations, like meaning, is this applicable to different populations? And if it doesn't meet the standards, as they are minimum standards that are in place for something to be considered for a drug. So if they don't meet those minimum standards, then there's fewer drugs and devices that get discovered. And so it's a whole snowball effect. Now, one of the challenges that people do have very low in certain groups is what they've noticed, particularly racial and ethnic minority groups, rural living groups, older adults, and people of a lower socioeconomic status. The racial component is actually very important, because by 2045, it's expected that less than half of the U.S. population will be white or Caucasian. So That's a problem. So some of the things that go into why people don't participate is centered around a concept called implicit bias, which we can go into a whole nother time. We can have a whole other discussion on. But implicit bias is something that creeps into clinical decision making, and a lot of people won't offer the patient of certain populations to participate. They'll assume that they're going to say no, or different things like that. So that's one area. But when minorities are offered the inadequate invitation or the opportunity, there are a number of barriers that have been determined that kind of continuously come up as for why they choose not to participate in recruitment or in being in the clinical trials. One of them has to do with some of the proximity to clinical trials. So the access to the research institutes. If you think about, some people don't even have a primary care doctor, don't even have health insurance. So then they're less likely to be around an institute that's doing that type of research to even be offered the clinical trial, or if they might live too far away from where the trial is taking place. Some of the things is just the consent forms can be very intimidating for some people, that either they can't have a lower reading level, or it might not be in their primary language that they speak. They might have some of these consent forms have a lot of medical jargon in them, and they're just way too long. So it's just very intimidating for a lot of people. And other people have trouble with transportation. So, if you have to take public transportation, or you don't have reliable transportation, then you're definitely not going to go out of your way to try to get to a clinical trial. But then there's also other things that are related to why people don't participate. Is there's something that people don't realize, is that you get compensated to be in clinical trials, but there's still a cost associated with being in them. If you have to take off time from work, then you're losing wages. So that's, some people in certain groups of people in minority populations tend to work in hourly wage type places where you don't get paid time off. So anytime you're choosing to request time off of work, then you're losing money. Or if you have to arrange childcare, those things are all deterrents for people. Or if you're sick and you have to withhold other treatments. Or, like I said, for certain studies, every study has eligibility criteria, and there's certain types of comorbidities or, pre-existing conditions, basically, that make it more challenging for diverse groups of people to participate, where certain diseases are heavily prevalent. So these things kind of all go into it, along with just the awareness. So unawareness of opportunities, or unaware of why participating in clinical trials are important, and language barriers, or inadequate explanation, or time to process information. I think certain populations that are hesitant, it's just they really just need time to digest the information. It's a lot of information at once, and people really just need to understand before they can comfortably make a decision. So those are types of things that, are a problem, but even if there are those things, there's also a lot of things that are surrounded itself around trust. There's a lot of mistrust. And that is completely it's completely warranted. Especially in certain groups, you always hear about Tuskegee syphilis study. So that's American, that's with African Americans specifically having, being abused. There's the Guatemala syphilis experiment. So that is Hispanic being abused. Henrietta Lacks is a big thing. That's that if there's a whole book, it's a movie now, and it's the immortal life of Henrietta Lacks. Henrietta Lacks was a woman who had cervical cancer, and they took samples of her cancer cells that were dividing, and they used that for research, and their research has lived on. It's been, they've used those cell lines for countless discoveries and things like that. And the family never even was told that they were taking her cells for research, and they never even have compensated her or anything. It's a very tragic study. And then there's the Havasupai, I'm sorry for mispronouncing it, the Native Americans, Indians. And there's also been some other types of just historical abuses. So every minority group has pretty much been abused in some kind of way by science. But I guess the light of the tunnel is that when these abuses did happen and are discovered, they've led to new guidelines put in place to protect populations from being abused. So I think the biggest thing, though, is people need to understand that people are weary of these things for a reason. There's also a lot of misinformation that's out there. And so there's a lot of concerns and issues around trust. And so science really has to work hard to build the trust of individuals and help people to become aware and to be transparent of these things, and have the appropriate amount of incentive and make it easier for people to participate.
Absolutely.
So yeah, it's a problem. So some of the things that people are trying to do to increase minority participation and participation overall is to foster these like relationships with people in the community. Because people might not trust science, but people might trust certain individuals. And so if you can form a relationship with the community, then that can really help with trust. But you have to also show the people in the community, like you have to be vetted by them. Like you can't just be like, oh, it's going to be fine. Like you really need to have a true partnership and really show like a commitment to this population and really kind of do some convincing there. But I think that adds like an extra layer of protection for the community as well. Also, there's just better materials and strategies. I think that a lot of times people are trying to like basically remarket things, because before these like things do not make you want to participate, because they'll just be like a random words on a page, and
as far as consent
attractive
forms and things like that.
Well, as far as like even like flyers and posters, like you might see like a random flyer, like, okay, this looks sketchy. Like it's not, something that's going to have people do that. But yeah, but then also having more methods for these consent types of documents, like having them in different languages and having people explain it and then have the person teach it back to you so that they can demonstrate their understanding. So I'll tell you about something, and then the patient would then teach it back to me so that I understand that they understand. Just kind of how we were talking about with the viruses, and you kind of told me your understanding and things like that. And then also just like making it easier to participate, like having it available during like after work hours, or offering childcare and things like that, or offering transportation. And also just around the staff, making sure that you're staffing people that are developing these trials, people that are working and administering these trials, that they represent people in the population that you're trying to get to participate. This is also the importance of why there needs to be more diversity in healthcare, and especially in as doctors and things like that, because the implicit bias that sets in, people might not even think of a study of coming with an idea of a study of something they want to study, because they might not realize that it's an issue, because they have these blind spots and they don't realize that like, oh, African Americans might deal with this differently. So there's different things like that all, play a role. And so it's really our job to really become educated, and to make sure meet my role as a physician in training and all of these things, as a scientist, is to really make sure that people do understand, because without that understanding, that's where the fear sets in. Like when you don't understand, that's where the fear comes in. And I think that people need to be compassionate with people that, I mean, obviously there are some people that do purposely, almost it seems like on purpose, so in misinformation and to try to cause discourse. But a lot of it is people that are trying to be well-meaning. People are really trying to figure out what's best for themselves and people are trying to weigh in like, okay, is it better for me to just get this, or should I risk the side effects of this vaccine and things like that? When like people don't want the cure to be worse than the actual disease itself. So
absolutely.
And actually, we didn't get to talk about the side effects and things like that of the.
Oh yeah, let's talk quickly about that. Yeah.
So
side effects.
So as far as the side effects for these vaccine, they're very typical of any vaccine. So typically around the day 2, some people on day 1, but usually around day 2 or 3, the majority of people had whatever side effects that they were going to be. And pretty much all of the side effects resolved. Like the side effects were almost non-existent. And these side effects were things like that basically tell you that your immune system is working, because they're signs of an immune response. So they would be fever, fatigue, and you get the fatigue because your body's putting in some extra work to try to make these new cells.
Body's putting in work. All right.
Headaches, because your blood vessels might be expanding to allow more cells to come through faster because they're packaging these new cells. So that can cause some pressure and headaches, chills, all of those different things that are related to your changing body temperature, muscle pain, joint pain, all of those types of things that can happen. The most common effect is fatigue. You just feel a little bit run down, and you feel a little bit just kind of like sick. But that usually lasted, let's see, on this chart, about usually like a day. Usually get over it in a day. So the most common like immediate type of thing is redness or swelling or pain at the injection site. So, I mean, that's just one of those things that can happen. These are very favorable. This is not unique to any vaccine and everything. And some people also bring up, like maybe they're not allergic to vaccines in general, but they are allergic. Maybe they're afraid that maybe they're allergic to something that is in the vaccines. One of the things that comes up as a misinformation is like, oh, well, there's mercury in vaccines, or oh, there's different things like that. Aside from the fact that this is a falsehood, this particular vaccine actually has no preservatives whatsoever. That's why it has to be stored in these crazy cold like freezers, because they're preservative free. Like I said, they are literally a bubble of fat with the